Okay, so good morning or good afternoon, everyone. My name is Dan Peer. I'm the Vice President for Research and Development at Tel Aviv University. I'm happy to open the second joint symposia between Tel Aviv University and Rutgers, joint scientific symposium, this time exploring drug discovery. And I want to thank Jack Barros for all his help. And to remind everyone that you can submit questions in the Q&A at the bottom of the screen. The symposium will be recorded and shared. And so I want to start by introducing Mr. Mark Levinson as the chair of New Jersey Israel Commission. So Mark S. Levinson serves as the chair of New Jersey Israel Commission, an appointment of the government of the state of New Jersey. Professionally, he is the chair of the real estate department and chair of the real estate transaction practice group at the law firm Sills, Commons and Gross, PC, located in Newark, New Jersey. He also chairs the firm's Israel Business Practice Group. Mr. Levinson is a long-standing member of the firm's management committee. He handles a broad range of transactions for domestic and international clients and had 37 years of experience in private practice, including 8 years as a senior real estate lawyer at the major financial services institution. During his 8-year tenure as in-house counsel, Mr. Levinson oversaw all domestic U.S. real estate matters and numerous worldwide real estate development and construction projects for his employer. Mr. Levinson also serves as the arbiter in New York City civil court system, as a mediator in the United States District Court for Southern District of New York mediation program, and a director of the New York chapter of the National Association of Corporate Real Estate Executives. He's a member of the American Bar Association Real Estate Properties Committee and served as the chairman, president, or as an executive committee member of many non-for-profit organizations. He received numerous awards for his volunteer activities and does significant professional pro bono work for various non-profit organizations such as A Better Chance, the Joseph Kushner Hebrew Academy, the Anti-Defamation League, the Jewish Federation of Greater DPhil, and the Zionist Organization of America. So I want to welcome him, and we will be happy to hear a few words of greeting from him. Thank you.
Thank you. It's my pleasure to be here at this symposium. The efforts that have gone underway to get this going, especially during the end of this pandemic, have really been remarkable. The relationship between the state of New Jersey and Tel Aviv University and the state of New Jersey, especially, can you hear me?
Yeah, we hear you well.
I'll just continue. I've been to the video disabled. So I'm sorry, I'll just rewind a bit. I'm Mark Levinson. I'm chair of the New Jersey Israel Commission, and it's my honor and privilege to have served as chair now for 10 years. And, under the governor, under the state of New Jersey, Governor Phil Murphy, who traveled and organized a trip to Israel 2 years ago, we really moved into first gear. The efforts to really have Rutgers and Tel Aviv University work together on something significant. New Jersey and Israel really have so much in common. As has been often said, we really are the same geographic size. We have the same amount of population, 8.5 million people. Both states really tremendous intellectual capabilities, creativity, great universities, great talent pool of workers. There's so much that these two states have in common, and Rutgers being really the flagship statewide university and system in New Jersey, and Tel Aviv being one of the most prominent universities, Tel Aviv University being one of the most prominent universities in the world. This really is a natural partnership. I couldn't be more pleased and proud of the effort that's been ongoing. It is really a tribute to both universities that during this pandemic, we've been able to establish. This is a second in an ongoing series of programs. And we are so looking forward to the end of the pandemic, to kind of the introduction of the vaccines into our collective systems. And we hope that next time we meet, if not next time, then maybe the second time, we hope to meet you in Tel Aviv at your beautiful, beautiful campuses, which, by the way, I want to point out that many of the buildings at the Tel Aviv University campus, which I've been to many times, have been sponsored or underwritten by major philanthropists from New Jersey. There's always been a very significant close relationship between the state of New Jersey, the citizens, the philanthropists in New Jersey, and Tel Aviv University. So this is really a natural fit. I very much enjoyed the opening program a few weeks ago. This program today will be great, and we really look forward to continuing not only contributions by both sides, but really growing to create one of the best collaborations that we hope people will see and be a model for future collaboration going forward. So with that, I really welcome everyone this morning, and I know that the program will be terrific, and I thank you all for joining us there, knowing it's late afternoon your time. Thank you very much.
Thank you so much, Mark. Okay, so I want to present our first speaker is Professor Ehud Gazit. Professor Gazit is an endowed chair at Tel Aviv University and member of the executive board of the university. In 2015, he was knighted by the Italian Republic for service to science and society. And Professor Gazit is a member of the National Council of Research and Development. From 2012 to 14, he served as the chief scientist of the Israeli Ministry of Science and Technology and the coordinator of the Forum of Chief Scientists of the Israeli Ministries. And from 2008 to 2012, he actually serves in my position. So he was the vice president for research and development and the chairman of the board of directors of Ramot, the technology transfer company of Tel Aviv University. Prior to this appointment as a vice president, he also serves in different academic and administrative positions at the university, including the head of the chemistry, biology, double major track, a member of the university committee for appointments and promotion, and the head of the academic committee of the Rich Institute for Nanobiology and Nanotechnology, and also as a member of and managing board of the Center for Nanoscience and Nanotechnology. So it's a pleasure to have him here as our second speaker in this series. And he will talk about the first Blavatnik Center for Drug Discovery, and then a little bit about his lab and his work. So thank you so much.
Thank you very much, Dan. So let me share the microphone here. Thank you very much for the very kind introduction. So I will tell you today, as we have limited time, about the Blavatnik Center for Drug Discovery, which was a decision made by myself after I ended my other roles, as Professor Dan Peer mentioned. I felt that drug discovery, and especially early stage drug discovery, is something that is needed in our university and the Israeli ecosystem. And our motto of the center is turning excellent multidisciplinary science into therapeutic leads, as you can see. For me, myself, I have my affiliations both in life science as well as engineering, and also in the Segal School of Neuroscience, which is a university-based center. So I started a little bit with, turning excellent multidisciplinary science to therapeutic leads, and my affiliations, as I mentioned earlier. I'll start with a little bit about my own research and how I relate multidisciplinary science into therapeutic leads. Our approach for many years was trying to identify the most fundamental recognition modules in nature. Our approach is reductionist, minimalistic, non-biased, meaning we take complex biological molecules and cut into smaller and smaller pieces to identify the most fundamental recognition modules that can form ordered assemblies. A great line of the work that is being done in the lab is related to material science, because these biological structures form all kinds of assemblies with unique physical properties. I call it a solid state biology and a reference to solid state physics to mention the fact that you get very interesting mechanical, optical, piezoelectric, semiconductive properties in this due to the arrangement of the molecules in the solid state. But today we are speaking about drug discovery and our understanding of the mechanism by which metabolites and peptides form ordered structures and way to control it. We developed various technological avenues towards therapeutics, and the most advanced one is a spin-off of Tel Aviv University, is Biomedic Therapeutics, which is already in entering phase 2 clinical trials for treating AMD, age-related macular degeneration, and Alzheimer's disease. And we have 2 products in the line. You are welcome to visit the website. And we have other projects that are in preclinical stage, and all of it is related to the way in which we understand molecular self-assembly and being able to control it, to manipulate it toward the development of drugs. So about the Blavatnik Center for Drug Discovery, this is a unique structure in which we have under one roof, we have a state-of-the-art environment, but leading to early stage drug discovery. Our aim and approach is to early stage. We take technology from the university, from researchers from the 17 affiliated hospitals with Tel Aviv University, and being able to bring it to a level either to license it to a pharma company, or to fund a spin-off company, or to mature the technology enough in order to bring it to the next step. And indeed, what we really do is combination with our multidisciplinary approach of chemistry, biology, and computer-assisted drug design, as I will mention in more details next. As I said, we provide the academic environment. We are a research institute. We are not only serving as a service or in-house service facility, but rather doing research. We also have internal research within the center, and we have to develop biological assays, chemical assays, and through optimization and target identification, as well as validation pipeline. We are well into translational research. We aim to discover one of the directions that we take. I will mention our different areas is to relate to the treatment of rare disease. This is something that is very important to us. Actually, we now serve as a hub for families that unfortunately have children with rare disease and no remedy, and we help them to identify either novel therapy or to go through some drug repurposing, and this is quite a major activity for our center. We do personalized medicine based on high throughput screening platform. We take patient samples. We work very closely with physicians in order to test various activity on cells from patients. We have automated infrastructure, state-of-the-art for doing this activity, and we promote and sponsor academic research, as I said, to reach industrial requirement. Many times we have excellent research coming from the university, yet the research may be not mature enough. The researcher may be excellent in basic research, but they are less familiar with the needed work and building portfolio that is required by pharmaceutical industry, and we help them with this method. We have a collaborative work with TU researchers from Tel Aviv University, as well as other institutes in Israel and also abroad. We don't have yet a project with New Jersey, but we will be very happy to have one. We provide a mechanism for in-kind financial support to test original research hypotheses. So we have a system in which researchers provide us with their ideas. Quite short and easy mechanism. It's been evaluated for its scientific excellence, as well as the opportunity and the chances to get into therapeutic leads, and we can provide some funding, in-kind funding. Our approach is to have the research done at our center and not to provide a lab with financial support that may go to other purposes. So we are very much focused, and thus we are very cost effective in the way that we develop this direction. We have dedicated research and development laboratories, and our approach is to develop small molecule drugs. We are not, as we have nice funding from Mr. Blavatnik, yet we are limited in our financial sources, and we decided to focus on small molecules, and this is what we help with. We have our academic, we have our leadership, but it's based on full-time employees of the center. They are running the everyday activity. I'm more like a chairman of the center, but we have a managing director, CSO, Dr. Ludmila Bujansky, Dr. Avi Rave, and we had different heads of the different units. We have a head of Elvira. Dr. Elvira Chaimov is the head of the medicinal chemistry unit. Dr. Edward Pinichuk is the head of the high throughput screening and biological assay unit. And we have Dr. Hamutal Engel, which is head of the computer assisted drug design. They are research associates within the university. They're running the everyday operation. They are involved in research, they are involved in innovation, but also involved in some services to industry, as I will mention later. We have also academic leaderships, Dr. Roy Amir, Professor Roy Amir, Professor Miguel Weil, Professor Chaim Wolfson, which are providing our faculty members within Tel Aviv University, and they provide us with academic guidance towards the activity that we do. We have a very strong international advisory board. This was my decision when I established the center to have very strong, opinionated, and very helpful scientific advisory board. I think that you can recognize some or many of the names that we have here. Leaders from academia, industry, in the United States, in Europe, in Israel, and before the COVID-19 epidemics, we had the pleasure to host the advisory board here. as many of the things we moved into the virtual forums, but still are extremely helpful in providing us with guidance, as well as helping and helping specific guidance for projects that we find as most close to their heart and scientific and industrial background. We have also an academic board, Professor from Tel Aviv University, which provides us with some board of directors, as you can see the corporate structure that we had here. Maybe it's the product of too many years in various capacities, as Dan mentioned. But I think that, and if my recommendation for a person who wants to establish a new center, it's very good to have these balances, the checks and balances, and strong boards, which help you to focus. And I think that you would see just a few years after establishing the center, we have quite a lot of products. Our areas of research, I mentioned already rare disease. We are very strong at metabolic disorders, inborn order of metabolism, neurodegenerative disorders, oncology, cardiology. We have quite a lot of research now related to ion channels in cardiology. It's about 75% of our project are in these 5 areas, but this is not because we decided that this is the areas. It was a bottom-up, grassroots type of activity in which we called for proposal from our faculty and affiliated hospitals, and being able to identify those areas in which we have a stronghold of Tel Aviv University. You already saw this slide, but you can see it in a larger magnification. So we have our approach to get novel therapeutics, and based on our interdisciplinary approach or multidisciplinary approach, indeed we use tools from biology, chemistry, and computer science. We're working extensively in high-throughput screening for target identification and validation. We use computer-assisted drug design for molecular design and target modeling. And we have also a medicinal chemistry unit. I mentioned Elvira, which we recruited from Teva. So this is something that you don't often find in academic institutes, but we do have a strong and very efficient unit that allow us to do the medicinal chemistry in lead design and synthesis, as well as structural activity and quantitative relationship. A little bit about each of the units. In the high throughputs unit, we are very strong in establishing new assays. We have researchers coming from all kinds of institutes and medical establishments, even some that have their own HTS coming to our unit. So people are very welcome to come and present the project. We are in the unit itself. We combine biology and computer science with very strong eye content analysis, image analysis, which is used for our some of our screening, in which we look at the properties of cells from patients, and we combine the technology, instrumentation, and most importantly, methodologies for some researchers who were never involved in such activities. As some of the assays that we do are related to cell-based assays, enzymatic and binding assays. We have all kinds of biophysical tools to study it, assays that are involved high content analysis, as I mentioned, and also we have a data scientist on the unit, which helps us a lot in the analysis of a large body of data and providing. So I move fast to the second unit, which is the computer-assisted drug design, in which we are using both commercial available software as well as our own research tools that were developed here, and we're doing molecular dynamics, structure-based drug design, legal-based drug design, and quite a lot of cheminformatics. In which is we are providing quite important data for our researchers, also those that are less familiar with computer-assisted drug design. Finally, as I mentioned, the medicinal chemistry unit is involved with synthesis of various molecules, including peptides, natural products, and classical medicinal chemistry activity. We have currently 38 projects. We have service projects, and we have projects with industrial partners in Israel, as well as international. As you will know, for choosing products, choosing projects for development are of course the scientific strength, which is it's a need, it's a must, but we're also looking for unmet clinical needs as well as commercial opportunities. We work very closely with our tech transfer company, and this provides us with helping to choose and nurture those projects that we would like to, that are appropriate for further development. We already have one spin-off company from the center. We have several collaborative projects that are funded by the Israel Innovation Authority. We have several patents as well as many publications. After all, we are a research center. We have many academic activity related to workshops, conferences. We have graduate courses, so and some hubs and training graduate students. So we see ourselves as a hybrid. On the one hand, of a center which provides a state-of-the-art technological activity. We provide financial means for early stage projects, but we also help with the academic activities. We have educational activities, several conferences that were held by the center. We had a New Horizon in Rare Disease, drug discovery in rare disease. Child health was another direction that we took, and we see ourselves also as a center, a hub for moving and promoting early stage drug discovery in Israel. We have, as I mentioned, also project with industry. As Dan mentioned, I was also in some government positions, and I see an importance for the ability to work closely with industry for the development of excellent therapeutic needs. And we are involved also in for example, in the Prime Minister's Innovation Summit, and we are very much into it. Just the last slide, Professor Pierre also gave me already the tell me that the time is off. We are donor, the Blavatnik Family Foundation found our research very advancing, and we got another donation which allow us to establish a metabolite medicine division for the center, which, as with the spirit of the center, we will combine metabolite profiling by advanced mass spectrometry, some of the mass spectrometer that are first installed in Israel for imaging mass spectrometry, and state-of-the-art activity, studying metabolite pharmacology, and also metaboloinformatics, which combination of computer science and metabolite studies. Thank you for your attention. It was very quick. You are very welcome to visit our website. Just type in the Google Blavatnik Center for Drug Discovery, and we also have a virtual tour in the center, so you can look around. Thank you very much.
Okay, so that's me this.
Yes, you need this.
And I would like, no problem.
Stop sharing. Stop sharing.
I would like to ask Dr. David Kimball to present our next speaker.
Thank you very much. Well, thank you, Dr. Gazit, for a really impressive presentation. And I must say, I'm looking forward to our future collaborations between Tel Aviv University and Rutgers, and in particular with the Blavatnik Center, because drug discovery and development is my personal dream and desire as well. So good afternoon and good morning. It is my great pleasure to be back for the second in our ongoing series of symposia and to introduce my friend and colleague, Dr. Stephen Burley. Stephen has a broad range of experience and expertise in structural and computational biology, in biophysics, structure-based drug discovery, and clinical medicine, particularly in oncology. Relevant to today's talk, he's the director of the RCSB Protein Data Bank, and within Rutgers University, he is university professor and Henry Rutgers Chair, the founding director of the Institute for Quantitative Biomedicine, and member of the Cancer Institute of New Jersey, where he co-leads the Cancer Pharmacology Research Program. Dr. Burley received his doctorate from Oxford, where he was Rhodes Scholar. Then he trained at Harvard Medical School for his MD. He became a Howard Hughes investigator at Rockefeller University, and from there made the transition to biotech to become CSO and senior vice president of SGX Pharmaceuticals in San Diego. And there he was responsible for leading the discovery and development of multiple anti-cancer drugs. Eventually, SGX was acquired by Lilly, and Stephen did spend a few years in Indianapolis before we were able to bring him to Rutgers University, where he's been since 2013. Dr. Burley's presentation today is titled Protein Data Bank, Impact on Drug Discovery and Early Drug Development. And Stephen, it's your...
Thank you very much, David. My thanks to, hold on a second, I'm just trying to find the, sorry, my thanks to Mr. Levinson and the leadership of Rutgers and Tel Aviv universities, and Dr. Gazit for leading off the program with a fascinating description of the Blavatnik Center. So my topic today is the Protein Data Bank. David enumerated my various roles at Rutgers. The one that requires most of my time, my day job, is running the Protein Data Bank for the United States government. So I will very, very briefly, I will touch on the history of the PDB. We are in our 50th year of operations, continuous operations, and then go on to describe how we support global research and education, and then specifically to talk about some recently published work enumerating the impact of the Protein Data Bank and structural biologists on drug discovery. And then I will change gears and talk about what we've done in the time of the COVID-19 pandemic to do our part in helping to overcome this global health crisis. And I'll finally I'll wrap up by acknowledging the very large team of people who make all this possible. The Protein Data Bank was established in 1971 as the first open access digital data resource in all of biology. It was founded with just 7 X-ray structures of proteins and has evolved to become the single global archive for protein, DNA, and RNA experimental 3-dimensional structures. Today, there are more than 170,000 structures, atomic level structures of macromolecules that are available open access from the PDB. Since 2003, the US has shared responsibilities for managing the PDB archive with data centers in Europe and Japan, including Protein Data Bank Europe in the UK and Protein Data Bank Japan at Osaka University. There's a longstanding connection between the PDB and Israeli scientists. Among the most prolific of depositors to the PDB is your Nobel laureate, Ada Yonath, who determined the structure of the ribosome. I should also mention that Dr. Joel Sussman was a director of the Protein Data Bank here in the United States for a time when it was at Brookhaven National Lab before it transitioned to Rutgers at the end of the 1990s. The RCSB PDB supports global research and education in fundamental biology, biomedicine, and the energy sciences. We integrate structural data from all areas of science, literally from agriculture to zoology, and validate, biocurate this information, archive it in the Protein Data Bank, and then make it available through our research website, rcsb.org. This is used by millions around the world in academia and in industry. Most of the large, if not all of the large biopharmaceutical companies maintain a copy of the Protein Data Bank archive inside their firewall to interoperate with proprietary structures that have been determined by crystallographers in the working within the company. As the PDB archive keeper within the Worldwide Protein Data Bank organization, the RCSB PDB has a special responsibility for safeguarding the archive, which has an estimated replacement cost today of about $17 billion US. My colleagues and I have recently published 2 papers analyzing in quantitative terms the impact of the Protein Data Bank on new drug approvals by the US Food and Drug Administration. First, in a paper published last year in Structure, we showed that discovery and early stage development of nearly 90% of the 210 new drugs approved by the US FDA between the beginning of 2010 and the end of 2016 was facilitated by open access to PDB structure data. The impact of these structures ranged all the way from target identification to structure-guided drug discovery, as exemplified by this structure of vemurafenib bound to a mutant form of the BRAF protein kinase that's abnormal in about 50% of patients with late-stage melanomas. The drug was approved subject to a clinical test, a genetic test, documenting the presence of the mutation in the patients. In a second paper published earlier this year in Drug Discovery Today, we examined the impact of the PDB on new anti-cancer drugs approved by US FDA 2010 through 2018. Discovery and development of approximately 90% of the 79 new anti-neoplastic drugs approved by US FDA was facilitated by open access to PDB data. We also showed that more than 70% of the 54 new small molecule anti-cancer drugs were the products of structure-guided drug discovery by drug hunting teams in industry labs. In most cases, the biopharmaceutical company benefited from open access to structures of the target proteins previously contributed by academic structural biologists. The Protein Data Bank is widely regarded as being comprehensive, authoritative, and of high scientific value. It has been certified by the Core Trust Seal as a trustworthy data repository and is regarded as a gold standard exemplar open access archive for biological data maintained as a public good. PDB impact on small molecule drug discovery and early development occurs at 5 stages in the process. Knowledge of 3D structures informs our understanding of the protein's role in disease, contributing to target validation. Close inspection of the target protein in 3D helps to identify the drug binding site, contributing to our understanding of druggability. Visualizing tool compounds bound in the active site or the drug binding site helps us understand the chemotype binding properties of the target protein. And finally, structure-guided drug discovery facilitates optimization of lead series for potency and selectivity during the medicinal chemistry campaign. Early stage development is also benefits from access to PDB data. Optimization of ADME properties can frequently be improved by looking at structures in the PDB, such as those of the cytochrome P450s, or the P-glycoprotein drug resistance pumps, which are responsible for pushing chemotherapeutic agents out of malignant cells. And to address safety concerns, there's a structure of the human ether agogo potassium channel in the PDB, with which binding to the channel can be assessed with the goal of avoiding QT prolongation and the risk of a lethal arrhythmia. Now let me move to SARS-CoV-2 and the timeline, our timeline for the pandemic. We all know that late in 2019, an unexplained influenza-like illness began to take hold in Wuhan in the People's Republic of China. In January 2020, the causative agent was revealed as a novel coronavirus dubbed SARS-CoV-2, and the genome sequence was released on January the 10th. Later in the month, a team from Shanghai Tech in the People's Republic of China contributed the first of the SARS-CoV-2 proteins to the protein data bank, that of the NSP5, or the main protease. It has a PDB ID code of 6 Lima uniform 7 to use the NATO alphabet. Tomorrow's total, when we do our weekly update, for the number of SARS-CoV-2 structures in the PDB will be a whopping 777. The unseen enemy, as you're all aware from this airborne illness, are the aerosol droplets containing the virus that are extruded by individuals who are not wearing masks. And you can see in this beautiful and haunting watercolor, reminiscent of 2001: A Space Odyssey, the virus, SARS-CoV-2, in a respiratory droplet surrounded by mucins and antibodies and other materials. The RCSB Protein Data Bank has been very hard at work during this time. We've prioritized biocuration of the incoming structures over all of our other activities. We have strongly encouraged all of our depositors to release their COVID-19 structures immediately, and I'm pleased to say that virtually all of our depositors, upon receiving that request, agreed to it. We're using a consistent taxonomy name for the virus and consistent UniProt referencing for the proteins. And you can find a summary of all of the SARS-CoV-2 proteins and related resources at this URL. For public education, we've also made a series of videos, one of which is shown here, explaining the importance of hand washing, one of the 3 key public health defenses that we can practice before the vaccines become generally available. That's masks, social distancing, and good personal hygiene. A bit about the SARS-CoV-2 genome. It's a single-stranded positive sense, 5' end capped and polyadenylated messenger RNA. It looks exactly like a cellular mRNA, which is one of the ways the virus eludes the body's immune system. Almost all of the non-structural proteins are expressed as two polyproteins, 1A and 1AB, depicted here. And they, like the HIV polyprotein, require cleavage by not one, but two viral proteases. The PL protease, papain-like protease, which cleaves here, here, and here to liberate the first three proteins from the two polyproteins, and then the 3C-like or main protease, NSP5, which liberates itself and a host of other polyproteins, many of which are drug discovery targets. A number of subgenomic RNAs are responsible for the expression of the structural proteins, the spike protein, the E protein, the M protein, and the nucleocapsid protein, plus a series of so-called open reading frame proteins, or ORFs. The NSP5 main protease was the first to enter the PDB. The archive now houses a large number of apo and co-crystal structures of the protein with as of last week, about 165 small molecule ligands. This particular rendition of the NSP5 main protease, a dimeric protein, is shown here with a covalently bound inhibitor sitting in the active site facing us, and you can just see a trace of the inhibitor in the active site of the second monomer, which is facing away from us. This is a chymotrypsin-like enzyme, as I mentioned earlier. It uses a cysteine-histidine catalytic dyad to cleave the viral polyprotein at 10 sites, releasing these non-structural proteins 4, 5, 6, 7, 8, 9, 10, 12, 13, 14, 15, and 16. Non-structural proteins 7, 8, and 12 make up the RNA-dependent RNA polymerase, which is the target of remdesivir, the only validated drug target in the genome of SARS-CoV-2. We recently undertook and have published on bioRxiv, and are currently working with a journal to get published a comprehensive discussion of SARS-CoV-2 protein evolution during the course of the pandemic. And I'll just give you one vignette from that study of 29 different viral proteins. For the non-structural protein NSP5, we analyzed 46,797 viral proteomes. 94% of these had NSP5 unchanged. The 6% where the viral protease was changed could be apportioned into 216 unique sequence variants. 189 were single mutant variants. There were 26 doubles and 2 triples. Most of the substitutions we saw in these unique sequence variants were non-conserved, 62% versus 38% conserved. We then went on to build structural models for all of the unique sequence variants, and determine that the sequence differences mapped to the core, the boundary region, and the surface in this proportion. So only a minority of the substitutions mapped to the core of the protein, and most of those were conservative. The boundary and surface regions of the protein on the middle and outer shell each had about 43% of the substitutions, and the bulk of these, more than 60%, were non-conservative. Most of the changes were marginally destabilizing. We did see one very interesting mutation in the active site affecting the catalytic dyad, changing this histidine to another residue, and we're in the process of evaluating that as an alternate version of the enzyme. We saw no significant impact on mutations in the dimer interface, and no other substantive changes in the active site residues. So the good news here is that this is an excellent drug discovery target. because it's undergone virtually no change in terms of the business end of the molecule during the course of the first 6 months of the pandemic. I want to close with a sobering postscript. The main proteases of coronaviruses are extremely highly conserved. And if we compare the amino acid sequence of the SARS-CoV-2 main protease with that of its SARS-CoV-1 counterpart, they're 95% identical. My company, SGX Pharmaceuticals, contributed the first structure of a SARS-CoV-1 protein to the PDB back in 2003. It's PDB-ID1 Quebec 2 whiskey. These two proteins, the two main proteases from SARS-CoV-1 and SARS-CoV-2, are extremely similar in three-dimensional structure. The root mean square deviation on C-alpha atomic coordinates is only 0.8 angstroms, less than the diameter of the hydrogen atom, and you can see the overlay of the two structures here. The SARS-CoV-2 is in gold, purple, and blue, and the SARS-CoV-1 is in green, solid green, and you can see that the two structures are virtually identical. When you look at the active site, all of the amino acids involved in binding this inhibitor are identical, and the root mean square deviation for all of the non-hydrogen atoms in the active site is less than half the diameter of a hydrogen atom. So what this tells us is a safe and effective drug against SARS NSP5 would have worked against SARS-CoV-2. And the bad news, the sad story here with the benefit of hindsight is that literally investment of a few $100 million in a SARS-CoV-1 main protease inhibitor would have saved millions of lives and trillions of dollars in economic losses worldwide. I did push in 2003 for a corporate sponsor of SGX Pharmaceuticals to prosecute a drug discovery campaign on the green protein here, which would have worked for SARS-CoV-2. Unfortunately, I was unsuccessful. An economically rational decision was made, but one that was, with the benefit of hindsight, of course, the wrong decision. So with that, I will acknowledge the contributions of the enormous PDB team that make it possible for me to stand up here today and talk to you and serve the interests of tens of thousands of structural biologists around the world and millions of PDB data consumers. We're funded by National Science Foundation, the National Institutes of Health, and the US Department of Energy. We're hosted at Rutgers principally, with a small contingent at UC San Diego at the supercomputer there, and an even smaller contingent at UCSF. I mentioned the Worldwide Protein Data Bank Partnership, which is crucial for the sustainability of the PDB. And ensuring that this remains a global open access data resource for the next 50 years. So with that, I will stop sharing, and I'll be very happy to answer any questions along with Dr. Gazit. Thank you.
Thank you, Stephen, for the great presentation. So I'm here to introduce Vince Smeraglia, who will handle the question and answer session. By way of introduction, Vince is the executive director at Rutgers of the Rutgers Core Facilities. He also is directing the rubric, or what are the Rutgers University Biomedical Research Cores. Those include, as with Blavatnik, medicinal chemists from industry, research pathology, and an imaging center. So we have also a very strong and abiding interest in drug discovery and drug development at Rutgers. And as I said before, I look forward to further collaborations. So Vince, please take the stage.
Thanks very much, David. And several questions have come in for Dr. Burley related to his last few slides, frankly, and what was done during the early parts of the COVID crisis to move forward with drug discovery as well as vaccine development. And some of the questions relate to when the Chinese research group initially published a sequence. Did they also cooperate and collaborate with the PDB to deposit relevant sequences of the virus, spike proteins, et cetera? And was there any interaction there? beyond deposits. If you could share with us a little bit about that time frame in January, February.
Sure, I'd be happy to do that. So the sequences, as we all know, was made public on the 10th of January, and that went into to a number of different nucleic acid databases around the world, and then that was immediately picked up by our collaborators Uniprot, and at NCBI at the National Library of Medicine here in the US, and all of the protein sequences were made publicly available from those two venues. In late January, we received the first deposition of a structure from the team at Shanghai Tech, and that was released on the 5th in our next available update cycle, which was on the 5th of February. We gave that particular structure absolute priority over all other structures that were due for release in the following week. And since then, we've seen a deluge of structures coming in from teams around the world. I think that the most important collaboration that we have in this regard is with the team at Diamond, the Diamond Synchrotron in the UK. We've received literally hundreds of new coronavirus protein structures with small molecule fragments bound to the active sites of various enzymes, and we prioritize those for validation, biocuration, and immediate release. And they're all now in the public domain. Every one of these structures of a fragment, a chemical moiety that has been essentially extracted from approved drugs, and made into chemical moieties, made into a library and used for screening. And these are all available, open access, no cost, and no limitations on usage. We've also been making these data available to our pharmaceutical company colleagues, who are a number of them are working on the main protease and the other protease, the papain-like protease. The structures of the spike protein were particularly important in the design of the new vaccines that have come out from both Pfizer and Moderna. Both of those. Both of the mRNAs that are used in those two vaccines contain changes that alter the amino acid sequence of the spike protein, incorporating two prolines at a key position in the protein. This locks the spike protein into a more immunogenic conformation, a more immunogenic shape, and that's probably one of the reasons why both the Moderna and the Pfizer vaccines showed such high efficacy when they did the initial readout from the phase 3 clinical trial. That information definitely guided the decision-making at the two companies, all open access information. I should say, to Pfizer's credit, they have actually determined the 3D structure of that modified spike protein and published that and made that available through the PDB to anybody who wants to try to improve on their design, because these are just the first generation vaccines. This is not the end of the story by any stretch of the imagination.
Thank you, Dr. Burley. And I know there are a number of folks on the webinar that are interested in not just the drug discovery science, but public policy, and a number of government officials. And this is not just a credit to you, but to everyone that interacts with the PDB and how it's helped guide us through this crisis. And frankly, the slide you showed on all the drug approvals is heartening as well. Those are taxpayer dollars at work, and it's impressive.
Indeed. Thank you, Vince.
Yeah. The next question actually comes from the Rutgers, a faculty member at the Rutgers, New Jersey Medical School, and I've actually seen a number of questions that are variations on this theme, and the question is for Dr. Gazit. Dr. Gazit, let's say hypothetically a Rutgers researcher at one of our medical schools wanted to interact with the Blavatnik Center at Tel Aviv University. Could you share with us what are the inputs and what are the sort of minimum requirements to have a solid and substantive discussion about a possible collaboration? For example, let's say the faculty member has an interesting target and a couple pro molecules, and they're thinking of doing a screen. They don't really know anything about high throughput screening. Do they, for example, need a robust assay with a robust fluorescent readout or some other readout to be able to talk to you about a project? In other words, what are the inputs that you're going to want to see in an initial discussion before you agree to take on a project?
Thank you very much for the question. It's given me an opportunity to elaborate on our process. As I mentioned in one of the slides, what we are looking for is a combination of excellent science. We look for unmet medical need, and as well as some commercial opportunity. And all of this is being done in close discussion with advisors that we have in our center, with our tech transfer company, and with members of our excellent scientific advisory board. As related to the maturity of the project, here is a decision that we made actually to allow researchers which are not in this field, which are newcomers, which do not know anything about high throughput screening, about the way to put up an assay, to get into this world. So if the science is excellent, it's unmet need, there's commercial opportunities, we would seriously consider the project, even if the researcher is his or her first time in developing a drug, so a drug candidate. And we are very open to discussion, we are very open to collaboration. Having said so, we also have projects that are much more advanced. We have people who are coming with actually already a lead, they want to optimize it, looking for changing in some properties. Classical medicinal chemistry, as many of you in the audience are familiar with. And again, this is something that those of you who came from industry or affiliated with industry, and this is quite common in academic environment, this is much less available to researchers, and we try to bring this. And in some cases, and I'm very happy to hear the excellent presentation of Dr. Burley. We are starting with protein structures. We have interesting targets. We're starting actually a virtual screen where you can screen millions of compounds. And then we are moving through the medicinal chemistry and the biological assay development. So as a short answer, we are open to those with not significant background or actually zero background in drug discovery if they have excellent science and unmet clinical need and commercial opportunities. It could be at very early stage, it could be in intermediate stage, and it could be actually in advanced stage in some of our faculty members who already had spin-off companies in which we further develop those leads to make them much more druggable and appropriate to enter into the clinics.
Thank you very much, Dr. That's an answer that's going to make our faculty members happy. That there's an open collaboration there. The next question actually goes to Dr. Burley, and it relates to several questions asked by corporate scientists, actually, biotech and pharma scientists. And that is essentially that they're aware not only that Rutgers oversees the protein data bank, but there's at the Proteomics Institute and other places at Rutgers, there's a certain amount of expertise in structural biology that could be brought to bear on biotech projects. And the question is, how does, if at all, how do biotechs and pharmaceutical companies approach you for collaboration? Are there annotated versions of the PDB? access to cryo-EM and other tools, how can they interact with the Proteomics Institute and with the PDB?
Thank you, Vince. So we, as our former president said, Rutgers is open for business. And we are very interested in fostering collaborations between the pharma industry and the Protein Data Bank, and the pharma industry and Rutgers scientists. We just for the avoidance of doubt, the Protein Data Bank contains a large number of cryo-EM structures of many different types of molecules, including the spike protein from SARS-CoV-2, and all of this information is of course publicly available at no charge and with no restrictions on usage. The cryo-EM, the cryo-electron nanoimaging facility which is located in the basement of the proteomics building, which is the headquarters of the PDB and the Institute for Quantitative Biomedicine. This facility is actively involved in collaborations with a number of different pharmaceutical companies, and I can put my email in the chat. Anybody who would like to write to me can do so, and I will be happy to connect you with the right person in the. I'm just typing it in right now. In the Rutgers system to do structural biology collaborations. All three of the major methods are represented at Rutgers. That's X-ray crystallography, NMR spectroscopy, and probably of most interest to pharma companies today, because they already have strong NMR and crystallography programs in-house, is cryo-electron microscopy. So all expertise in all three of those techniques is present at Rutgers in the labs of excellent faculty members. In addition to single particle cryo-electron microscopy, we also have a specialist in cryo-electron tomography, which allows visualization of molecular machines that work inside cells. Probably not relevant for drug discovery today, but very much so for understanding target biology and target druggability.
Thanks, Dr. Burley.
And of course, we work closely with Dr. Kimball and his team, including Vince, to make any collaboration with industry a success.
Thank you for adding that. I will also pile on a bit and say that the medicinal chemistry group that I have the privilege of overseeing not only works with many faculty at Rutgers and other universities, but spends a lot of time interacting with corporate partners and working on sort of being the virtual drug discovery arm for biotech companies in San Francisco and other places. The next question seems to be directed to Gazit. Both of you can feel free to answer. And that's what it comes down to is whether it's a faculty member at Rutgers or Tel Aviv University, or whether it's a small company, there are challenges in securing the funding to do drug discovery research. Although it's changed quite a bit in the United States over the last 25 years, it's not the first priority of funding for the National Institutes of Health and other funding organizations. And I wonder how you deal with bootstrapping projects when there are only small amounts of money. Does your Blavatnik Center have access to capital? Do you partner with other groups to bring forward the most promising projects? What would you say about the financial inputs to getting projects done?
Okay, thank you for this very important question. Indeed, this is a major obstacle for academics trying to fund early stage drug discovery. As I said, this was the reason that we decided to establish the center. Therefore, you need to be very precise and very efficient in the allocation of funds. As I said, unlike other mechanisms with funding is not transferred from the boundaries of the Blavatnik Center. So we provide researchers with the accessibility to state-of-the-art equipment, with state-of-the-art thinking and methodologies, but we do not provide funding for their own labs, which makes it, many of us are academics, and not always you are spending the entire allocated research grant in a very directed way. So in this case, we provide an in-kind support for those researchers. In some cases, and this is an Israeli initiative, as I mentioned, we're getting funding from the Israel Innovation Authority, which used to be the Office of the Chief Scientist, which is a mechanism that the government of Israel had established to support early stage of industrial development, not only in pharma, but also in other domains. And we already have several projects that are related to this. And actually the Innovation Authority likes it very much because we provide something that is closer to what we are used to in dealing with spin-off company or startup companies, but still in the backyard of the academic institute, enjoying all the benefits, as all of us know, also of being in an academic institute, not only in terms of science, and this is important, it's a very important process in which you are interacting with experts in the field, but also other infrastructures that you have in the university, instrumental research infrastructure, that for a small startup company or a spin-off of the university, it's much less available. We are working very closely with our tech transfer company, which has its own funding, some seed funding mechanism, but we are also looking for partnerships, and we are open for a dialogue in terms of having a collaboration with both governmental agencies as well as private equity. And we are, as I said, we are well familiar with the transfer of technology, first of all, the maturation of technology to the level that is attractive to external bodies, but also in the transferring process and in collaboration with the university and the tech transfer company. So this is where we stand.
Thank you. That's quite helpful. And I will second some of the remarks you made at the tail end there, that a lot of the collaborations our MedChem group ends up engaging in are those where we are a co-PI on a grant application, or we participate in the United States. It's called the SBIR program, where the federal government provides resources to smaller companies, which then can sub-award to universities to do translational work. So there are mechanisms to bootstrap projects when there's not a lot of funding there to begin with.
If I may add, it's for us, it's also quite challenging because if you look at the classical academic grants, they usually are less fond of having a person who is affiliated with the center and going very much into applied science and not a regular faculty member. But we're trying to deal with this as well.
Yeah, I think we say the same challenges here in the US with the NIH and the NSF, which is why they put together the SBIR program. But the dollar amounts there are modest initially. Once you've shown success, of course, the dollar amounts can be very substantial.
That's exactly right, yeah. I'll switch to you, Dr. Burley, for this next question. much of the work that you do and your colleagues do there at the Proteomics Center can be described into two buckets, some of which is clearly pre-competitive research, and then other parts of your capabilities tend to be translational and may, through collaboration, end up generating and creating substantive intellectual property. And I guess I wonder, this question keeps coming up with our corporate colleagues, although both you and I and Dr. Kimball spend a lot of time talking about this subject. How does a university, how does a research program and a proteomics center, for example, deal with intellectual property? And do you try to bifurcate pre-competitive work with the more translational aspects where IP can be created? How do you engage with, say, startup companies who have a concern about intellectual property?
So I think the approach to the pre-competitive information is simple. Anything that is released by the Protein Data Bank is regarded as a public good and is clearly identified as public domain pre-competitive information. I will say that we rely on Vince, on you and David and your office when contracts are put together with industry to arrive at an equitable approach to identifying areas where IP can be created, and then how inventorship should be adjudicated. It's at the end of the day, inventorship is a matter of law. It's not a matter of opinion, and it's important to get things right at the time of establishing a contract. And I'm having been married in a previous life to a lawyer, no longer. I know that. One one who's not skilled in that area shouldn't pretend to understand it, and we defer to you. And as the former President Bocci said, Rutgers is open for business, and the leadership of the Office of Research and Economic Development under David exemplifies that spirit. And as do we in the Institute for Quantitative Biomedicine and in the core facilities that are in the proteomics building in the basement.
And I just want to add that the New Jersey Israel Commission stands ready, willing, and able to help on either side of the equation or on both sides to help move anything and everything forward. And
Thank you. Thank you. And I think Mark would say this as well. I think flexibility around these issues is important when you're collaborating with a startup. Dr. Gazit, do you, not to sort of pin you down on intellectual property, but do you have any broad notions of how you handle intellectual property if the partnership or collaboration you're entering into is, for example, a startup biotech company rather than a pure academic collaboration? What are your thoughts on intellectual property?
So I have to say, until this moment, our collaborations were either internal or with other non-profit organizations, as hospitals, universities in Israel, outside of Israel. Usually we deal with startup or established company. This is more for service, and I have to say honestly that it's also a source for income for the center. So we can then fund all these nice things that you asked me in the previous question. Yet, we are in discussions in some models to have shared IP with entities, especially those entities that are a span of Tel Aviv University. I think that our tech transfer company is maybe this this may be one of the next presentation that we want from Tel Aviv University. Is a very efficient, very much oriented toward the researchers and find reasonable solutions. Of course, they protect the IP of Tel Aviv University. This is their role. they handle our portfolio, but they do find logical solutions in some cases. They're tough negotiator, which is good for us in some cases, but we also have a very reasonable solution. And in the past, we found some solutions like this. And I should say, Blavatnik Center is a center of Tel Aviv University. So our IP is assigned to Tel Aviv University, to promote. In Israel, it's where Dr. Burley mentioned lawyers. In Israel, as you may know, we don't have tech transfer office, but rather tech transfer company, which is a for-profit company, which is held, in the case of Tel Aviv University, fully owned by a non-profit organization, which makes our lawyer happy because there are some considerations. This is mainly related to taxation. But we have our tech transfer company. We have our IP assigned to the tech transfer company, and we are goes by the bylaws of Tel Aviv University regarding patents. But answering your questions, in the case of Blavatnik, we didn't have this yet. We had some discussions, but in the terms of Tel Aviv University at large, the portfolio is of thousands of patents. There were solutions in the past that were found.
Right. Thank you. Yeah, I notice Israel is somewhat like aspects of the United Kingdom, where the commercialization arm is a for-profit corporation rather than part of the academic institution, and that creates certain flexibilities and in terms of interacting with outside corporate partners and taking equity positions. A number of different things open up when your commercialization arm is a separate and apart for-profit company. It looks like we're starting to run out of time. I want to end things here by thanking Mark Levinson for bringing us together, for Dan Peer and Dr. David Kimball for spearheading the last few seminars at Symposium. There are many to come going forward. Of course, to Stephen Burley and to Ehud Gazit for those wonderful presentations and seeds of potential collaboration between Rutgers on the one hand and Tel Aviv University on the other. There will be, just a general announcement, there will be another symposium that will come to us in mid to late January, and we will provide information. All the folks who are participants on this meeting as well as the prior meeting, we will send out a web link to let you know about the details of the next symposium co-hosted by Tel Aviv University and Rutgers University. And I thank you all for your participation. Thank you for your questions. Again, to Dr. Gazit and Dr. Burley for those very helpful presentations and remarks. And look forward to seeing you all at the next Rutgers Tel Aviv University Research Symposium. Thank you all very much.
Thank you, Vince. Thank you, Ehud.
Terrific program. Thank you, gentlemen.
Thank you, Mr. Levinson.